is Zoey Nichols. I am a junior here at McDaniel College. I am a biology major with a molecular specialization. And here at McDaniel, I am a member of the women's golf team, as well as the president of Alpha Sigma Tau.
Hi everyone, my name is Jayce Klingenberg. I am a senior here at McDaniel College with a double major in health sciences and dual chemistry kinesiology. I am also a member of the women's basketball team and honors club. This summer, alongside a few other students and Zoey, we were given the opportunity of a lifetime, the chance to impact and make a difference during a pandemic that has left many people worried and hopeless. We were fortunate enough to have the opportunity to design and synthesize inhibitors of the SARS CoV2 main protease. Of course, everyone knows about SARS CoV2, commonly known as COVID-19 or coronavirus. This pandemic has made endless amount of impacts on everyone's daily lives and is continuing to alter how we go about things, whether it is work, school, sports, or even just socializing. While masks and other precautionary measures have limited the number of cases, we have still seen over 1 million deaths worldwide and over 35 million cases. To date, there is still no vaccine or antiviral drug to keep these numbers from climbing. SARS-CoV-2 is the distant relative of SARS-CoV that broke out just 17 years ago in China. Unfortunately for us this time around, SARS-CoV-2 is more virulent and has spread much faster. As a result, endless amount of funds, resources, and scientific minds have delved into the challenge of finding a vaccine. Unlike many of the researchers who are focused on the vaccine, companies like Diamond and Enamine have teamed up to release the crystallization of 66 fragments of the SARS-CoV-2 main protease. Unlike many other studies, this information was released to the public in hopes to find an antiviral drug at an accelerated rate during these unprecedented times. And this was an opportunity we could not give up. Vaccines have been unsuccessful in the past, like in the case of HIV. Antivirals have been an extremely effective alternative. Using similar methods, we began targeting the life cycle of the virus. Like many other viruses, the virus attaches to a membrane receptor to enter the cell. The genetic material is then transferred into polyproteins. Our goal this summer was to inhibit the cleaving of the protease so they could not reach the prior step. This would ultimately end the life cycle of the virus, which is what we were working to achieve all summer. The arrow in this image where the inhibition would occur, as you can see, if the virus is inhibited at that point, it will be unable to replicate and spread throughout our bodies. To find the potential antiviral drug, we looked into different optimization strategies. Looking further into our target, the chemotype of compounds were fished out, and we found 3 areas for optimization: the sulfonamide group, piperazine group, and chloroacetyl group. When we were able to covalently link different warheads to look for different routes for optimization. Essentially, our synthetic purpose was to simply attach different compounds that were readily and commercially available and see what worked. We had 3 basic synthetic routes that carried us throughout our research. Synthesis of the warhead replacement using tosyl chloride. Tosyl chloride was one of the initial hits that was found and was also readily available and affordable. Therefore, we attached it in our first synthesis. The goal was to see which, if any, of the warheads was most effective. Our next synthesis was for piperazine isoesters. Tosyl chloride was added to the diamine. The diamine was deprotected, and then chloroacetyl chloride was added. Our last involved the synthesis of the sulfonamide replacements. Our first step was Michael's addition, then deprotection of the Bach group, and lastly warhead substitution. Each of these steps were repeated and varied with only small changes in an attempt to find a compound with an effective % inhibition against the main protease.
We found that replacing the warheads with the following groups proved largely unsuccessful against both proteases. Tosylate groups are very commercially accessible, so we began with this approach. Because such low inhibition rates were found, we eventually decided to veer away from this scheme. Piperazine replacements were also not as effective as anticipated, although the inhibition percentages of the last two compounds on this chart should be acknowledged. Zach and Priscilla created very similar compounds with aromatic rings attached that did in fact yield high success rates against the main protease. Sulfonamide replacements gave us the most promising results. As Max in our lab was finding success with naptoquinones, we decided to try modifying these compounds. What we found was high inhibition rates against both proteases. Acetyl chloride was easily attached to the piperazine ring and found to be active not only alone, but also with substitutions such as that of compound 448. Based on our findings and the findings of others in our lab, in the future, further optimization of naptoquinones may lead to more success. Substituting various R groups in the above position is the main focus of further study. It may also be interesting to attach different groups to the linked naptoquinone and piperazine rings, such as replacing the acetyl group with other functional groups. As Jayce mentioned in the introduction, this year has been very hectic and called for a lot of adjusting. Typically, we would be standing in front of you with a big physical copy of this poster. As everyone is saying, these times are unprecedented. So here we are, and displayed now is all of the information we just presented to you together, all included on this one poster. I would like to thank my family, my friends, Dr. Ferraris, and my peers for helping me go through this experience. And this amazing opportunity also would not have been possible without the support of the McDaniel College Summer Research Fund, the Jean and Donald Richards Student Research Fund, McDaniel College Student Research and Creativity Grant, and the BSE Biosciences.
I would also like to say thank you to my peers, family, the chemistry department, and Dr. Ferraris for making this opportunity one to remember. I would also like to give special thanks to the Schofield Fund, the Don Fund, the Smith Fund, and the Hopkins Fund for making this opportunity possible.